Welcome back to the deep dive. Sometimes the most profound insights really come from looking at the smallest details.
Absolutely.
Like, take the doobie mouse, for example. This isn't just, any old lab mouse.
No, not at all.
It's this genetically engineered superstar, basically. Scientists actually called it synaptically smarter.
And that highlights something crucial.
Exactly. What makes it so special, it really all comes down to the incredible power of those tiny junctions in its brain. The synapses. And crucially, their amazing ability to change.
That plasticity, yeah.
So today we're doing a deep dive into synapses, these fundamental connections that let neurons talk to each other.
The very basis of nervous systems.
Right. And learning, memory, all of it. If you think of your brain like this, I don't know, incredibly complex computer,
Right.
the axons, the long nerve fibers are like the wires carrying signals. But the real magic, the sort of logical junctions that decide how well the whole thing performs, how it improves, that's the synapses.
That's a great analogy.
So our mission today, let's unpack how these tiny connections work, how they send signals, and maybe the most fascinating part, how they actually change over time, how we learn and remember things.
Yeah, how the brain rewires itself.
Exactly. So let's get connected and see how it all works.
Okay, so at its core, a synapse is basically a specialized contact site. It's where one neuron, the presynaptic one,
The sender.
right, the sender, influences another cell. That could be another neuron or maybe a muscle cell, the postsynaptic cell or effector.
Okay.
And what's really key to understand, like you said, is that usually they're not physically touching. There's this tiny gap.
The synaptic cleft.
Exactly, the synaptic cleft, usually around 20 to 30 nanometers
Yeah.
wide. It's across that little space that all this complex communication has to happen.
And when we talk about getting signals across that gap, there are really two main ways, aren't there? Electrical and chemical.
That's right. Sometimes called the Sparks versus Soups idea from way back.
Yeah, so electrical first. The Starks.
Yep, electrical synapses, these are the speed demons. Here, electric currents flow directly, like straight from one cell into the next. Instantly changes the membrane potential of the second cell.
How does that work with the gap?
Well, the gap is much smaller here, only about 3.5 nanometers.
Mhm.
And it's bridged by these special protein channels. They form structures called gap junctions.
So like little tunnels connecting them.
Precisely. Little tunnels made of proteins called connexons in us vertebrates or innexons in invertebrates. They create these low-resistance pathways for the current.
So instantaneous transmission, no delay, and often bidirectional, the signal can go both ways.
Often, yes. Super efficient.
Where do you need that kind of speed in nature? What's the advantage?
Well, their adaptive significance is all about speed or synchronization. Think about escape reflexes.
Right, like getting away from a predator.
Exactly. In crayfish or fish escape systems, every millisecond counts. Electrical synapses give them that edge. Or think about synchronizing lots of neurons firing together, like in fish that produce electric shocks. They need all those electric organ cells to fire at precisely the same instant.
Okay, that makes sense. Fast and coordinated. But then you have the chemical synapses. You said they're more common, even though they have that bigger gap. How do they bridge that gap if there's no direct connection? It seems, well, slower.
It is slower, definitely.
Yeah.
But that's where the trade-offs come in, the advantages. Chemical synapses work by converting that electrical signal from the first neuron,
The action potential.
right, into a chemical signal. The presynaptic neuron releases these chemical messengers, neurotransmitters.
Okay.
These molecules then diffuse. They just drift across that synaptic cleft. It happens fast, but it's still diffusion.
And then they hit the other side.
Yep. They bind to specific receptor proteins on the postsynaptic membrane. And that binding triggers a new electrical or sometimes a chemical change in the receiving cell. Structurally, they look different too. You see lots of tiny bubbles, vesicles, filled with neurotransmitters on the sending side.
Ready to release.
Exactly. And a dense patch of those receptor proteins on the receiving side, often on these little bumps called dendritic spines.
So yeah, it does sound more complicated and definitely slower than just a direct electrical zap. Why are they the majority then? What are these big advantages?
Well, there are several key ones. First, amplification.
Amplification.
Yeah. A single incoming electrical signal, one action potential, can cause the release of thousands of neurotransmitter molecules.
Wow.
And those thousands of molecules can open up potentially thousands of ion channels on the postsynaptic side. So a small input signal gets significantly boosted.
Okay, that's a big deal. What else?
Second, diversity of effect. Electrical synapses are almost always excitatory. They make the next cell more likely to fire.
Right.
Chemical synapses, though, they can be either excitatory or inhibitory. They can make the next cell less likely to fire. That gives the nervous system much finer control, like having both an accelerator and a brake.
Ah, inhibition. That's crucial. Okay, third.
Third is directionality. The signal pretty much always goes one way, from presynaptic to postsynaptic. It ensures information flows in the right direction through circuits.
Makes sense. And the fourth advantage, you mentioned this earlier, and it seems huge.
Yes, the fourth, and arguably the most profound, is plasticity.
Changeability.
Exactly. Chemical synapses are much, much more modifiable than electrical ones. Their strengths can change. They can get stronger if they're used a lot, or weaker if they're not used.
And that's the link to learning and memory.
Precisely. This inherent plasticity, this ability to change connection strength, is fundamental. It's how our nervous systems develop, how circuits fine-tune themselves, and critically, how we learn and remember things throughout our lives. It's how experience shapes the brain.
Okay, that is fascinating. So let's dig into that chemical transmission process, that intricate dance, as you called it. Walk us through the cycle. How does the neurotransmitter actually get released?
Okay, so it all kicks off when an action potential, that electrical spike, travels down the axon and arrives at the presynaptic terminal.
The end of the sending neuron.
Right. That arrival, that change in voltage, triggers the opening of specific ion channels, voltage-gated calcium channels.
Calcium. Okay, so calcium ions, C2+, rush into the terminal.
Exactly. Calcium floods in. And this influx of calcium is the critical trigger for the next step. People actually visualized this beautifully in early experiments, using things like jellyfish proteins that glow when calcium is present, especially in the giant synapses of squid, which are huge and easier to study.
So the calcium comes in, and that's the signal to release the neurotransmitter.
Yes, that calcium surge triggers a process called exocytosis. Those little vesicles we mentioned, packed with neurotransmitter, they move to the edge of the neuron, fuse with the presynaptic membrane, and basically spill their contents out into the synaptic cleft.
And it's not like one molecule at a time, you said?
No, that's another fascinating part. It's released in standardized packets called quanta. Each quantum is basically the contents of one synaptic vesicle, maybe around 5,000 neurotransmitter molecules.
Quanta, like little prepackaged signal units. How do we know it works like that? Is there experimental proof?
Oh, absolutely. Really elegant experiments. If you record from, say, a muscle cell that receives input from a neuron, even with no stimulation, you see these tiny, spontaneous little blips of depolarization.
Mini potentials.
Yeah, exactly. Miniature end plate potentials, or MEPSPs. Each one is tiny, maybe 0.4 millivolts. That's the response to one quantum, one vesicle's worth, being released spontaneously.
Okay.
Then, researchers cleverly manipulated the conditions like lowering the calcium concentration outside the neuron, which makes release less likely. When they did stimulate the neuron under these conditions, the evoked responses weren't smooth. They came in distinct steps.
Blips.
Yeah, the responses were always multiples of that basic 0.4 milliver Phi size, either 0.4 or 0.8 or 1.2 millivolts and so on, never in between. It showed that release is fundamentally quantal. It happens in these discrete packets, each corresponding to one vesicle fusing.
That's incredibly precise, like digital communication almost, in packets. And what happens to the vesicle membrane after it fuses and releases its contents? It doesn't just build up, right?
No, it absolutely has to be recycled. The cell membrane would just keep getting bigger otherwise.
Ah, right.
So after fusion, that vesicular membrane is rapidly retrieved back into the presynaptic terminal through endocytosis. It's pinched off and reformed into new vesicles, ready to be refilled with neurotransmitter.
Efficient recycling.
Very. There are a couple ways it happens, like a full fusion and retrieval, or sometimes a quick kiss and run, where the vesicle just opens a pore briefly. It involves a whole suite of specialized proteins like snares for docking the vesicle and synaptotagmin acting as a calcium sensor to trigger fusion. It's a complex molecular machine working incredibly fast.
Amazing molecular machinery. Okay, so the neurotransmitter released in quanta diffuses across the cleft. What happens when it reaches the postsynaptic side? How does that translate into excitation or inhibition?
It binds to those specific receptor proteins embedded in the postsynaptic membrane. And this binding causes ion channels to open or close, changing the flow of ions across the membrane.
Leading to EPSPs or IPSPs.
Exactly. If the effect is to make the inside of the cell less negative, closer to the threshold for firing an action potential, that's depolarization. We call that an excitatory postsynaptic potential, or EPSP.
Making it more likely to fire. How does that usually happen?
Typically, it involves opening channels that let positive ions, mainly sodium, A+, flow into the cell. Sometimes potassium K+ flows out too, but the net effect drives the membrane potential towards about 0 millivolts, which is depolarizing.
And you mentioned these can add up. Summation.
Yes, absolutely crucial. A single EPSP is usually tiny, far too small to trigger an action potential on its own, but they can summate. Temporal summation is when you get rapid, repeated firing from one synapse. The little EPSPs overlap in time and add up.
Okay.
And spatial summation is when multiple different synapses, perhaps on different parts of the neuron's dendrites, fire at roughly the same time. Their individual EPSPs spread and combine at the neuron's integration zone.
Like adding votes together.
Kind of, yeah. Think about a motor neuron in your spinal cord. It might receive inputs from like 10,000 other neurons.
Wow.
Each input is weak, but if enough excitatory inputs arrive close together in space or time, their combined effect can push the neuron over the threshold to fire its own action potential.
Okay, that's excitation. What about the flip side, inhibition, the IPSPs?
Right, inhibitory postsynaptic potentials, or IPSPs. These make the inside of the cell more negative or hyperpolarized, moving it away from the firing threshold.
Making it less likely to fire.
Exactly. This often happens by opening channels for negatively charged chloride ions, Cl, to flow into the cell, or sometimes by letting positive potassium ions, K+, flow out.
So the neuron is constantly doing this math, essentially, adding up all the EPSPs and subtracting all the IPSPs.
You could think of it that way. The neuron's output, whether it fires or not, and how often is basically an integral function of all these competing inputs arriving moment by moment. And importantly, where the synapse is located matters. Synapses closer to the axon hillock, that's the spot near the cell body, where the action potential is usually initiated, tend to have a bigger influence than synapses far out on the dendrites, because their signal degrades less as it travels. Location, location, location.
Fascinating. Okay, now you mentioned receptors, and
Yeah.
that there are two main kinds that the neurotransmitter binds to. What are they?
Right, two main classes. The first are ionotropic receptors.
Ionotropic.
Yeah, these are the simple, direct ones. They're essentially receptor channels.
Yep.
The protein that binds the neurotransmitter is the ion channel itself.
So binding directly opens the gate.
Exactly. Neurotransmitter binds, channel pops open, ions flow. It's very fast. The delay is minimal, like fractions of a millisecond. The classic example is the nicotinic acetylcholine receptor at the neuromuscular junction. Acetylcholine binds, channel opens, sodium rushes in, muscle contracts. Simple, direct, fast.
Got it. Direct action. What's the other class, though?
The second class is metabotropic receptors. These are quite different.
Metabotropic. Sounds more complex.
They are. These receptors are not ion channels themselves. Instead, they're usually G protein-coupled receptors, or GPCRs, big proteins that snake through the membrane 7 times.
Okay, so what happens when the neurotransmitter binds to them if they aren't channels?
Well, the action is indirect and usually slower, but often much longer lasting in its effects. When the neurotransmitter binds, it activates an intermediary molecule inside the cell called a G protein.
A middleman.
Yeah. This activated G protein then goes off and triggers a whole cascade of events inside the cell, what we call a signal transduction pathway. This often involves generating second messengers.
Second messengers, like internal signals.
Exactly. Molecules like cyclic AMP, CMP, or calcium ions themselves, or others like DAG and IP3. These second messengers then spread within the cell and activate other enzymes, often protein kinases.
Kinases, they add phosphate groups to things, right?
Right. And phosphorylating proteins can change their activity dramatically. So, for example, a neurotransmitter, like norepinephrine, might bind its metabotropic receptor, activate a G protein, which activates an enzyme called adenyl cyclase, which makes CAMP. CAM then activates a protein kinase, which can then phosphorylate various target proteins.
And those targets could be?
All sorts of things. They could be ion channels, so the metabotropic receptor indirectly opens or closes channels. For example, a type of acetylcholine receptor called muscarinic slows down heart muscle this way. But the targets could also be metabolic enzymes, changing the cell's energy use, or even proteins that regulate gene expression in the nucleus.
Wow, so these metabotropic receptors can have really broad, long-lasting effects way beyond just a quick blip in membrane potential. They can actually change the cell's fundamental state or behavior?
Absolutely. That slow, modulatory, far-reaching action is key to their function, especially in things like mood, attention, and, importantly, learning and memory, where you need lasting changes. They can even act presynaptically, maybe reducing calcium entry in another axon terminal to inhibit its neurotransmitter release.
Presynaptic inhibition. Okay. And what about the neurotransmitters themselves? There must be lots of different kinds.
Oh, dozens have been identified. We can broadly group them into small molecule transmitters, things like acetylcholine, amines like dopamine and serotonin, amino acids like glutamate, usually excitatory, and GABA, or glycine, usually inhibitory,
Okay.
and then larger neuropeptides. And it's not always just one type per neuron. Sometimes a single neuron can release more than one kind of neurotransmitter from the same terminal that's called co-transmission. Maybe a fast, small molecule and a slower-acting peptide.
Adding another layer of complexity. And you said earlier the same neurotransmitter can do different things.
Yes, this is super important. The effect doesn't depend just on the neurotransmitter chemical itself, but critically on the type of receptor it binds to on the postsynaptic cell.
Ah, the lock and key idea again.
Exactly. Take acetylcholine, ACH. At your neuromuscular junction, it binds to nicotinic ionotropic receptors, causing excitation and muscle contraction. Fast, direct.
Right.
But in your heart muscle, the same AC binds to muscarinic metabotropic receptors, initiating that G protein cascade we talked about, which ultimately leads to inhibition, slowing your heart rate. Same chemical, different receptor, opposite effect.
That's amazing. It really highlights the specificity of the system. So how does the signal stop? The neurotransmitter can't just hang around in the cleft forever.
No,
right?
definitely not. The action has to be brief and precisely timed for effective communication. It's terminated very quickly, usually within milliseconds.
Wow.
Primarily two ways. One is enzymatic degradation. An enzyme floating in the synaptic cleft literally chops up the neurotransmitter. The classic example is acetylcholine esterase breaking down acetylcholine. The other main way is reuptake. Specific transporter proteins, usually on the presynaptic terminal membrane or sometimes on nearby glial cells, actively pump the neurotransmitter back out of the cleft.
Recycling it.
Exactly. This happens for neurotransmitters like norepinephrine, dopamine, serotonin, and the amino acid transmitters. This rapid removal clears the cleft so the postsynaptic neuron can receive the next signal cleanly. It's essential for maintaining fidelity.
Okay, fantastic overview of the mechanics. We've got the structure, the electrical versus chemical, the release, the receptors, the termination. Now let's really connect this back to how our brains learn and remember. Back to the Doogie mouse idea and this concept of synaptic plasticity.
Yes, synaptic plasticity. It's just this idea, this fundamental property, that the strength or effectiveness of these synaptic connections can change over time based on patterns of activity.
Not fixed, but dynamic.
Exactly. And this change in synaptic strength is widely believed to be the cellular mechanism underlying how our nervous system adapts, learns, and stores memories.
So it's not just an on-off switch, but like a volume dial that can be turned up or down based on experience. Are there different types of this plasticity, different timescales?
Yes, definitely. We see short-term plasticity first. Things like synaptic facilitation, where if you stimulate a synapse repeatedly in quick succession, the response gets slightly bigger with each pulse.
Okay.
Or the opposite, synaptic depression or anti-facilitation, where the response gets weaker with repeated stimulation. And then there's post-tetanic potentiation, PTP, where a brief burst of really intense high-frequency stimulation causes a temporary but prolonged enhancement of the synaptic response that can last for minutes, maybe even hours.
So even on short timescales, synapses are constantly adjusting their gain.
Right, it shows their efficacy isn't fixed, it's dynamically regulated by recent activity.
And for understanding how these changes relate to actual learning, especially at a basic level, people often turn to simpler animal models, right? Like Eric Kandel's work with the sea slug, Aplysia, that won him a Nobel Prize.
It did, and it's truly iconic work, a cornerstone of learning and memory research. Aplysia has this simple reflex. If you touch its siphon, it withdraws its gill for protection.
Okay, a basic defensive reflex.
Right. Kandel and his colleagues showed how this simple behavior could be modified through experience, and they tracked down the synaptic changes responsible. For instance, if you repeatedly touch the siphon gently, the gill withdrawal response gets weaker and weaker. It habituates.
Habituation, getting used to it. What's happening at the synapse?
They found it was due to synaptic depression specifically. The sensory neuron that detects the touch releases less neurotransmitter onto the motor neuron that controls the gill.
Right.
Less signal gets through, probably due to less calcium influx with repeated stimulation.
Okay, but then they showed the opposite too, right? Sensitization?
Yes, sensitization. If you give the Aplysia an unrelated noxious stimulus, like a mild shock to its head or tail, and then touch its siphon again, the gill withdrawal response is now much stronger, much more vigorous than it was initially. The animal is sensitized, on alert.
And the synaptic basis for that.
That turned out to be presynaptic facilitation. The shock activates other neurons, interneurons, that release serotonin onto the presynaptic terminals of the sensory neuron.
Serotonin, okay.
The serotonin binds to metabotropic receptors on the sensory terminal, triggering that CAMP second messenger pathway we discussed. This leads, through protein kinases, to the phosphorylation of certain potassium channels.
Changing the channels.
Yeah, closing some potassium channels actually prolongs the duration of the action potential when it arrives at the terminal. A longer action potential means the voltage-gated calcium channels stay open longer.
More calcium influx.
Exactly. More calcium influx means more neurotransmitter vesicles fuse, releasing more neurotransmitter. So the synapse is strengthened presynaptic facilitation.
That's a beautiful molecular pathway linking experience to synaptic change, and Aplysia even showed how these could become long-term memories.
It did. That short-term sensitization based on modifying existing proteins lasts maybe an hour. But if you give the Aplysia repeated training sessions with the shock, you can induce long-term sensitization that lasts for days or weeks. And crucially, Candle showed that this long-term memory requires new protein synthesis. It involves activating gene expression in the nucleus, leading to the growth of new synaptic connections sometimes. It links short-term functional changes to long-term structural changes.
From sea slugs to mammals, let's talk about long-term potentiation, or LTP. Especially in the hippocampus, a brain region we know is vital for things like spatial learning and memory formation in us.
Right. LTP is perhaps the most studied form of synaptic plasticity in the mammalian brain, and it's widely considered a leading candidate mechanism for how we store certain types of memories.
What is it exactly?
It's a long-lasting increase in synaptic strength, specifically in the size of the postsynaptic response that's induced by brief, intense, high-frequency stimulation of the presynaptic pathway. It's like that post-tetanic potentiation we mentioned, but much, much more persistent. It can last for hours in brain slices and even weeks or months in living animals.
And it has this property related to timing, doesn't it? The Hebbian idea.
Yes, LTP is often described as Hebbian, named after Donald Hebb, who proposed that neurons that fire together wire together.
Okay.
LTP typically requires that the presynaptic neuron fires and the postsynaptic neuron is strongly activated, depolarized at the same time. It's associative. It strengthens connections that are active concurrently, which makes intuitive sense for learning associations.
So what's happening at the molecular level during LTP? Who are the key players, especially at those excitatory glutamate synapses in the hippocampus?
There are two crucial types of glutamate receptors involved here. First, AMPA receptors. These mediate the normal, fast excitatory transmission. Glutamate binds, they open, sodium flows in, you get a quick EPSP.
Standard excitation.
Right. But the second type, NMDA receptors, are the real key to inducing LTP. They're very special.
How so?
Well, they're often called molecular coincidence detectors. For an NMDA receptor channel to open and let ions through, two things need to happen simultaneously. First, glutamate must bind to the receptor, just like with AMPA receptors. But second, the postsynaptic membrane must already be strongly depolarized.
Why?
Because at normal resting potential, the NMDA channel is blocked by a magnesium ion, Mg2+, sitting inside it. Only when the cell depolarizes significantly, usually due to strong activation via nearby AMPA receptors, is that magnesium block expelled.
Ah, so it needs both the chemical signal, glutamate, and the strong electrical signal, depolarization, at the same time to open. Coincidence detection.
Exactly. And when it does open, it allows not only sodium, but also a significant amount of calcium, Ca2+, to enter the postsynaptic neuron.
Calcium again, the trigger.
Yes, that influx of calcium through the NMDA receptor acts as the critical second messenger signal that initiates the changes leading to LTP. It activates various calcium-dependent enzymes inside the spine, like CAMKII, calcium calmodulin-dependent protein kinase 2, and protein kinase C.
And what do those enzymes do to actually strengthen the synapse long term? How is LTP maintained?
A key mechanism for the expression or maintenance of LTP involves those AMPA receptors. The calcium influx triggers signaling cascades that lead to the insertion of more AMPA receptors into the postsynaptic membrane at that specific synapse.
More receptors on the surface.
Right. More AMPA receptors mean that the next time glutamate is released from the presynaptic terminal, it can open more channels, leading to a larger influx of sodium and thus a bigger EPSP. The synapse has become more sensitive, potentiated. They might also phosphorylate existing AMPA receptors to make them work better.
So strengthening the connection by adding more listeners, essentially.
That's a good way to put it, more sensitive listeners.
And if you can strengthen synapses, can you also weaken them? Is there an opposite process?
Yes, absolutely. It's called long-term depression, or LTD. It's a long-lasting decrease in synaptic strength. And interestingly, in the hippocampus, LTD induction also often depends on those same NMDA receptors.
The same ones. How does that work?
It seems to depend on the pattern and amount of calcium entry. The current thinking is that a large, rapid influx of calcium through NMDA receptors, like during high-frequency stimulation, triggers the kinases that lead to LTP. But a smaller, more prolonged, modest rise in calcium, like during low-frequency stimulation, might preferentially activate different enzymes, specifically phosphatases, which remove phosphate groups.
Okay.
And these phosphatases can lead to the removal or internalization of AMPA receptors from the synaptic membrane. Fewer receptors means weaker synaptic transmission, LTD.
So the same key receptor, NMDA, can mediate both strengthening and weakening, depending on the pattern of activity and the resulting calcium signal.
Yeah.
That allows for bidirectional plasticity.
Precisely. You need both to sculpt circuits effectively. You need to be able to strengthen important connections and weaken unimportant ones.
This raises a really important question, though. How do these molecular changes, adding or removing receptors, become truly permanent memories? Does the physical structure of the synapse change too?
That's a fantastic question, and the answer seems to be yes. This brings us to structural plasticity. Remember those dendritic spines we mentioned where most excitatory synapses are located?
Yeah, the little mushroom-shaped bumps.
Well, studies using advanced microscopy have shown that stimuli that induce LTP can actually cause the heads of these spines to grow larger, physically bigger, within minutes to hours.
The spines themselves change shape.
Yes. And even more strikingly, LTP-inducing protocols, or even just uncaging glutamate right onto a dendrite, can trigger the formation of entirely new dendritic spines, potentially forming new synaptic contacts. It's not just about changing the molecular components, the physical structure remodels too.
That's incredible. The brain is physically rewiring itself in response to activity patterns that might represent learning. Is there evidence of this happening in, like, a living animal that's actually learning something?
Absolutely. There's been remarkable work, for instance by researchers like Carol Svoboda, using two-photon microscopy to repeatedly image the same dendritic spines in the cortex of living mice over days and weeks, while the animals learn new tasks or experience changes in their sensory environment.
What did they see?
They saw exactly that. When the mouses experienced change, say, they trimmed some whiskers, altering sensory input, there was a dynamic turnover of spines. Some existing spines were eliminated, and new spines formed and became stable. This strongly suggests that learning involves the formation and stabilization of new synaptic connections and the elimination of others, literally reshaping the brain's circuits based on experience. This is a key adaptive strategy seen across the animal kingdom.
Wow. Okay, so this brings us full circle back to the doogie mouse. How does that specific example tie LTP directly to enhanced learning?
Well, the doogie mouse provides really compelling evidence. Joe Zine and his group engineered mice to overexpress a specific subunit of the NMDA receptor, one that's normally more prevalent in young animals and tends to keep the channel open a bit longer when activated.
Longer opening means?
More calcium influx for a given stimulus. And just as predicted, these doogie mice showed enhanced LTP in the hippocampus. Their synapses strengthened more easily and stayed strong for longer.
Okay, so they had boosted LTP. Did they actually learn better?
Yes, that was the crucial finding. These mice performed significantly better than normal mice on several learning and memory tasks, especially spatial learning tasks known to depend on the hippocampus. They remembered novel objects for longer, navigated mazes better. It showed that genetically enhancing LTP could directly enhance cognitive abilities.
That's pretty direct evidence linking the molecular mechanism LTP to the behavioral outcome, learning.
It is. And alongside that, studies that block LTP either pharmacologically or by knocking out genes essential for it, like the NMDA receptor itself or CAMKII, consistently show impairments in spatial learning. And more recently, work by people like Mark Baer has provided even more direct evidence.
Yeah.
They've managed to show that when an animal actually learns a specific task, you can subsequently go into its brain and detect the physiological signatures of LTP having occurred in the relevant circuits, using the same molecular pathways, like AMPA receptor insertion, that are involved in experimentally induced LTP. It strongly confirms that LTP isn't just an experimental curiosity. It's likely a fundamental mechanism the brain actually uses for learning and memory.
So let's try to wrap this up. We've journeyed from the basic definition of a synapse through the speed of electrical connections, the complexity and adaptability of chemical transmission,
Right, the quanta, the receptors, the second messengers.
and landed on this incredible concept of synaptic plasticity, from short-term tweaks to long-term potentiation and depression, potentially underpinned by actual structural changes in the brain.
Yeah, it's quite a story. And what's truly fascinating, I think, is realizing that these microscopic junctions, billions of them in our brains, aren't static components like in a fixed circuit board.
No, definitely not.
They are constantly dynamically changing,
Hmm.
adapting their strength, literally reshaping themselves based on every experience we have, everything we learn. This sort of staggering expansion in our understanding of plasticity shows just how incredibly dynamic our brains are, always learning, always rewiring.
It really is mind-boggling. So maybe a final thought for everyone listening. Next time you learn something new, whether it's a fact, a skill, or even just recognizing a face, pause for a second and consider what's happening inside your head. Millions, maybe billions of these tiny synaptic connections are subtly, or perhaps even dramatically, altering their properties. They're strengthening, weakening, maybe even physically changing, literally making you, your brain, a slightly different, more informed, more adapted version of yourself than you were moments before. What does this all mean for the future, for understanding the mind, maybe even enhancing it? It's certainly something incredible to think about.